I'm Jie Yang, and I'd like to talk to you about not really QSAR, but it's a catchy name of QSAR 21, but I really am going to talk about the chemistry. So the outline that I like to cover is how does chemistry help? How can it help? And I would like to give an example with a practical approach that we have taken, and that is implemented at the US FDA SIPSN, what we call mode of action QSAR, and the weight of evidence approach to put them all together. Then at the end, what they told me that they like to have. I'm going to start out a very simple, boring slide for you that Richard Dawkins said biology is so incredibly complex. And when I say that, when a person like that says, I should just go home. I don't know anything about biology, and it's really daunting. And so how do we actually come out of this very complex observations that we have? This is actually taken by the Fisher exact test of a Toxicity phase 1, 309 chemicals of in vivo experiments to in vitro experiments, and all those without doing any chemistry. So I was pretty discouraged by that. Next thing is then, since I'm a chemist, I thought maybe, is it possible that I remove certain chemicals that are very reactive? And is it possible that I actually start bringing some chemical groups and classes and slicing? Then it looks like there's some hope that I could actually see some correlations. So based on that, I'm going to start this talk and addressing the chemical characterization, what we call a chemical inherency, starting from the intrinsic properties of if I'm a chemical, by right I have certain properties and I have my right to have those properties. I'm a chemical, I could interact with another chemical, I have a reactivity, and among other chemicals also I could actually start exerting my reactivity to a biological system, start metabolic potentials. We could think about that. But it's still, these ranges are still intrinsic properties of the chemicals, and they are unsupervised, not supervised by the biology. And this second step is actually start linking the chemical intrinsic properties to the biological properties. And the third step would be the supervised, biology-supervised level that this chemical is associated now with either biological activity or toxicity. And so from then we start talking about structural alerts and rules. And at the end, the exposure in use cases that will actually define what is our risk and safety assessment should be. So this is the basic concept that I'm going to take you through, and I'm going to use like any knife or scissors I have to cut this cake. I'm going to look at it as a cake of my chemical space, and I'm going to slice and dice so that it could help me understand biology. So there is a role of this understanding huge inventory of the data in systems biology. We look at the chemical structures are related to the in vivo, in vitro experiments. And now we are trying to understand in vivo to in vitro, these relationships. Can we actually interject chemistry in order to do this? Sorry. So we're using, again, chemical structures to slice and dice, just another way of looking at things. And Richard and I have been thinking about this, and then we kind of collaborate of how we actually approach this. So I'm going to take a very practical example of Tox 21 inventory as a chemical inventory, and I'm going to slice and dice and see whether we really could use this method to get to where we want to understand how we could have a handle over biology. So the three big sources of that information, the inventory that we have, is from, of course, US EPA. Many of them are pesticides, but also they have now failed drugs that donated by the pharmaceuticals, and the green chemistry set, and the HPV and MPV. And also there are, I like to point out that there are some food chemicals from the US FDA CFSAN, and this is actually an example of how personal level of collaboration made this happen rather than the agency to agency, because I just sent it over to Anne and we got 100 compounds into that plate. Then there is NCGC plate, and they are mostly drugs, as you could see, from various sources of the information. Then we have NTP, of course, that are all the in vivo experiments that they have done. So these are all together had about 8,200 compounds. And now I'm start slicing in using different kind of knives, like I told you. Now this time I'm slicing by their chemical roles in use condition. So these are drugs, the biocides, and the food chemicals, and the cosmetics, and some general industrial chemicals and solvents and so forth. So of those inventory, Tox21, we have a large number of them are pharmaceutical, more than almost 4,000 compounds, about 3,800 compounds are pharmaceutical, and then about 900 were biocides. These are pesticides, herbicides, and all together I call it biocides. Then there's food chemicals. These are actually food ingredients and food contact substances, both direct and indirect. Then also the cosmetics ingredients and cosmetic-related chemicals. So this is really very important for us because as someone mentioned this morning, actually Derek Knight mentioned this morning that we have now the CIRAD consortium, and we are part of this COSMOS consortium that trying to come up with a innovative computational methods to provide an alternative for the repeat dose toxicity. And so we have actually put together a cosmetics inventory from COSING and PCPC, US and EU side. And we found about 1,500 compounds in this Tox21, so which means that we're going to get a lot of good assay data from it. So then we're going to start looking at what are the makeups of this chemistry. So I changed my knife now from a chemical roles of in-use case. Now I have a structural classes. I'm using that as my knife right now, and I'm going to slice the cake of 3 different NCGC plate and EPA and NTP. And as you can see, each individual structural fragment names are not that important here. What's important is that these lines, the longer the lines are, there are more compounds. So this is actually the percent frequency in the data set in log scale. And if you see nothing in there, it means that it's empty. So you could see NCGC plate has several places that are empty, and they are quite complementing between NCGC and EPA, and also NTP. So what this slide is telling you is that by combining these inventories and combining these different chemicals, we are now actually able to accommodate more space and actually enrich the features. And another thing that's important is that not just that enriching the neighborhood, but also increasing the diversity. So here is the diversity. Another way to actually cut it is that one before was actually I'm looking at just an inventory. Now I'm looking at chemical roles against the same structural classes. So I'm looking at the drugs and biocides and food ingredients and additives and cosmetics. As you can see, they are, again, drugs are providing a whole lot of features, and that's what drugs are good for, because they have more complex compounds. And these, where the biocides lag is the benzothiazine and the beta-lactam and the carbohydrates. Biocides do not provide any of these features where the drug covers and the food and cosmetic covers. And you can see in the food chemicals, you shouldn't have any carbamates in food, and that's a good story. And we should not have any purines and steroids and pyrazoles in food, and we don't see that. And so this is why combining different inventories and different chemical roles in use cases and expanding our chemical space is so important. So now let's take a look at then. I'm using those features, structural features that I called structural fragment library. I'm using those as my comparison. So these are my knives, okay? And I'm using them to start clustering different parts of this chemical space. And so we often do this by either clustering, hierarchical clustering technique, or the principal component analysis. And as you can see, that the drugs and the cosmetics are very far off, okay? So the drugs are all up front in this part, and the green is the cosmetics, they are behind in a 3D, so you could see actually better in the 2D projections that the green and red, they never actually overlap. The black and gray is the biocides, and blue is the food, and food and cosmetics actually have a lot of more overlap, and then the drug and the biocide have more overlap. As you can see, the heterocycles, and they actually overlap quite a bit in drugs and biocide, and the food and cosmetics are also, because of the dyes and the antimicrobials and things like that, so they actually have more commonalities. Then I'm comparing. So I just want you to understand that what I'm doing first going by the structural features against the chemical roles and against the different inventories. Now I'm using physical-chemical properties. And when we use physical-chemical properties of this, the log P water solubility, dipole moment, and that I listed over here, that I could very clearly separate between the drugs and cosmetics again. So it was, again, whether I use structural features or physical-chemical properties, those drugs and cosmetics are in different space. An important part is that by testing both of them, that we are now able to reflect the properties, intrinsic properties, the nature of the chemical from very different groups, and cover more. And to show you that, I'm just going to go very quickly, take a look at this. This is you could look at it as almost like a chromatogram of a compound just splitting up. In between, that one on the y-axis is actually using a structural features as hierarchical clustering, and the x-axis I use the physical-chemical properties, and the gray dot are chemicals, and in this case, is biocide. And I'm going to add drugs in here. That's what you see. And as you have seen, the drugs and biocide, although similar, they are going to actually expand the feature space and the physical-chemical property space, and you see that. The next thing I'm going to add is food and cosmetics. So it's actually filling up. And if we do this right and design it correctly, I think we may be just able to cover enough chemical space with certain limited number of compounds. And that is actually the whole thing about chemoinformatics being used for the drug design and chemical design and green chemistry design, being able to fill out this space. Now, the next step I'm doing is I'm actually skipping a couple steps of the metabolism, the reactivity. I'm going to actually toxicity. So I have looked for all those in vivo data for those 8,000 compounds. Of course, we don't have any of that, all of that, but I was able to find about 2,000 to 3,000 compounds, which I use normally as my training set in my models. But using those actual data, the toxicity data of 7 endpoints. So 7 endpoints are here: the salmonella mutagenicity and the rat and mouse tumorigenicity, and some developmental effects. And I'm using those to cluster compounds. And again, so this axis was again, y-axis was the structural feature clustering, and x-axis is the toxicity clustering. And this color are representing the different types of food or cosmetics or drugs. Okay, and now we begin to see some hopes of where things happening. So some of the structures you could see and where they show, and so the next point I like to take it to is from this information of structural classes and the features and where it actually related to the toxic endpoints, this is how we actually pull out knowledge, structural knowledge. So some of that is represented here. The top part is actually my cleft palate alerts for the steroid biosynthesis inhibitors and the retinoids and things like that. So you could see on the top part is a cleft palate, and the down in this the famous aromatic nitriles and the epoxide and the Michael acceptors and alkyl halides, the reactive alkyl halides, and these things are well-known genotoxic carcinogens. And we could actually slice and dice again the Tox21 and see how many percentage of the chemicals are actually alerted for genotoxic carcinogens. So in terms of the drugs, you can see the drugs and biocides and food and chemicals and cosmetics. It's interesting that actually some of those food actually still shows the BSI and the cleft palate alerts. And there are some interesting things that I like to talk to you about a little later. So this is a step that we actually start learning as a informatician. We're learning from the data. We're looking at the data with different features, with a different feature set from the structure or the properties, and we're slicing and dicing a different way, and we are building knowledge. And that's what I just showed you, how we actually go about it and doing that. And some of the endpoints are simple enough or well understood enough, where the chemistry role is so strong that we are able to understand this before and being able to model this even. So some of those we call mode of action models that are better actually understood is skin sensitization. Derek mentioned it today. And then the genotoxic carcinogens, these are also better understood in the genetic toxicity. So I'm taking it back where we have a better handle over biology is where we know chemistry a little better, how the chemistry works with the biology. So I'm just taking back to you this chemical inherency, and I'm going to actually start talking about this part, the biology and the predictions. So what is mode of action pathways and what is AOP? And this is really very complex, and I would not rather go with that. But what I understand, one thing from my perspective is I need something very practical, knife, that I could cut this very complex biology through. So as you can see, simple retinoic acid or simple chemical will first interact with the first event, with the biological target, and we call that molecular initiating event. That's going to trigger a whole network of responses, of cellular responses, which will eventually manifest as phenotypic effects in the organs, and then perhaps eventually appear as adverse outcome to the population. Whatever that is, which the first part where that molecular initiating event, when that is very well understood, and that related to this phenotypic effect, then we could start talking about mode of action categories, the chemical categories. So let's take a look at this simple retinoic acid, or likes of retinoic acid, what it does to this cleft palate. So first what I do is then take that as mode of action category, take it through this biological network, and find related chemicals through the pathway, and that's how I actually build a training set for that particular phenotypic effects. And the next set that I'm showing you is that, like I said, there are some very well-known approaches with, for example, like the genotoxic carcinogens and things like that. So we know the mutagenicity and clastogenicity and how it actually appear as the tumorigenicity. And through this, we understand pretty well. So again, the point is, if we understand the in vitro to the in vivo, if we want to actually connect that. Now, most of us actually at this in this workshop in systems biology, you are working on this biological features. You are concentrating on biological models. And as refreshing as beautiful this is, I just like to point out that how chemists failed while we're doing a QSAR is because we completely ignored biology. If you completely ignore chemistry, the consequence may be the same. So what I like to propose at this point is let's not repeat our chemist fault, our mistakes, that we do better this time, that use biological and chemical features together, okay, and cut through this mode of action categories, or whatever you call, use this knowledge, and if you have those knowledge, please share and teach us how to do that. Sorry. So in this case, I'm just using as an example of cleft palate as a practical example, and I'm taking this certain chemical classes, and I've gone through these pathways, and I found that yes, it's involving Wnt and the sonic hedgehog and the growth factor, those pathways, and so I'm looking at certain interactions, and I found chemicals that are doing those things. So these are actually my chemical classes right there. All those, related to those, to that key events that could lead to this effect on these pathways, and then eventually a disruption in the epithelium, the cell proliferation, and the palate closure. Okay, so that when the fusion doesn't happen, and this is what we see as a phenotype effect. So that was what we modeled. While we're doing that, how actually we found out what is related to what is where you have to actually use ontology to pull that data mining information. So I'm just going to go through very quickly that ontology is not a big thing. It's just we use these terms that to relate. For example, I have this, the phenotypic effects go through this, it happens in a site. Okay? Eventually, it's going to have to come through the cell, and cell is where things are meet. The chemistry and biology meet in the cell. And so biology pathway-wise, the proteins and genes are going to be explained. Okay. From the chemistry, also has its pathways. It has reactivities, and it has all properties that it goes through. So we have to understand both things, and it's going to meet in the cell, and that's where we have to actually pay attention to. And that is what we actually put together as an ontology, how we actually data mine those. Okay. So this data set was put together from the literature and from the regulatory database, because I have been working actually at the FDA, CIFSEN, for a couple of years as a sabbatical. So I have the data from the FDA, and of course ToxFDP from EPA was very useful. And then I was part of this IRC developmental database, so I had all the sources. And it's not just getting the compound and biology yes or no data. We have to have all the way down to the dose level through the ontology, so that I could actually go specific targeted effects that I could pull out. Okay. And those effects were related in maternal tox, or the fetal survival and growth, or the fetal morphology. And this time I'm looking for in particular for the cleft palate. So through this exercise, we found these categories, which you will recognize right away, and we will know where it actually hits. And those what I call a cleft palate alerts are down here, and these are more general ones on the top. And I'm actually showing if it's orange, it's actually up. If it's green, it's less toxic. So he has a. I just aggregated for your own viewing. So the maternal, the fetal, and the red morphology of cleft palate, and the rabbit, and this one is morphology is actually aggregated for the chronofacial, and the cleft palate is by itself. And also what we have done is to actually pull the metabolism active features. And I don't have time to go over this one now, but this is something that chemoinformatics should be able to help you to understand the metabolism better. So these are the ones that I used to further take into models. And when we do these models, then it's just a pure chemistry that we use mechanistic descriptors, the structures, and the physicochemical properties. And also the possible is that we could use QHTS data, the ToxCast data, or the TOX21 data. For example, in this case, the TGF-beta1 was used. So here is the scheme for mode of action, and there are several mode of action models, so it has to be put together. And the way that we put together is through by the weight of evidence approach using the Bayesian. So I would say if I have this chemical in a query, then it comes through, it hit alert right there. There is a triazole with the sigma charge positive on the carbon, and the mode of action category is going to triazole. And in this case, nothing else, but the TGF-beta1 is calculated, and the global was also calculated, and there's an equation for getting a weight for that. And at the end, you get this models, not just for each individual mode of action models, but at the end, you will get a weight of evidence combined model for concordant sensitivity and specificity. You could calculate many different ways. But important part is that these combined models based on weight of evidence is much more robust than by itself, the models by themselves. So the summary of this is that my really message is that I'm begging the biologists not to forget chemistry, because if we do, then the outcome will be not very much new learning. And this linking chemistry to the biology through this mode of action categories and use these chemical categories to explore the biology could really help. I'd just like to share quickly with you that what we have done in CIFSEN is that we have interviewed many toxicologists and reviewers for the system that they would like to have, what kind of system they would like to use. And this is a very difficult area because it's a very complex data, and at least 4 or 5 different kind of methods are all thrown at them, like structural alerts, the reader cross, and the threshold of toxicological concerns, and the cues of predictions. There are so many of them, they don't know what to do with it. So through our interviews, what we found out is that they would like to know what chemicals are actually reviewed, by when and how, what the status is, and about that decision, they want to know the clear mechanistic understanding of that. And they would like to have make a decision on a support system, but they don't like to make it harder because they don't have time to learn a new system. And sifting through so many of this information is more difficult than actually not. I wouldn't go there. So what we actually come up with is that they would like to have the transparent system. We're working on that. And then they actually accept the systems biology approach quite well, as long as it's actionable and pragmatic. And they would like to put the quantitative weight of evidence method in the computational method. And this dashboard and war room and red flags, and these are all their paradigm to make things easy.
Thank you. Chihei, do you want to stay for a couple of questions just to see if we have any questions? The microphone is not on, so.
I'm going to go back to sort of the beginning of your talk to one of the points I think you tried to make, and that was that for us to do QSAR, we have to have a rich enough chemical space to be confident that we can derive the relationships we want for the unknown compounds we're going to test. I think that was one of the early points that you tried to make, which rings home with me. And it made me think about something, the analysis that you did in terms of presenting the chemical space. I would argue, as a biologist and a toxicologist, that if we think about the future for this Tox21 approach, an in vitro approach to toxicology, one can also imagine it just like having a chemical space, that you have to have a cellular space. And let me explain what I mean. Let's assume there's 1,000 cell types in the body that are all important for one toxicity or another. Let's further assume that there's 1,000 kinds of interactions between some of those. For example, interactions between elements of the endocrine system, between specific cell types. So now you've got a matrix that's basically 2 dimensions and 1,000,000 boxes. That if you're going to have confidence doing toxicology by in vitro systems with some cells, and not all, I wonder what an analysis of the biological space, the cellular space, and the cell interactome, if you want to call it that space, would look like, right? Is there 50 cell types we have to have full coverage and confidence we'll hit all the mechanisms we need to, or is there 10, or is there 1,000? And where are we now in the Tox21 types of programs, wherever they are, and how do we get to a place where we have that kind of rich coverage? Could you imagine actually doing that kind of analysis for that space? That's my question to you.
Actually, yes, that's a very good question, because ever since the Toxcast phase 1, I think many of us actually looked at that data, I'm sure. Everybody did that, right? And tried to see, was it really necessary to run all the 600 endpoints, 600 HTS assays? How are they related? Can we actually, are there certain things are actually correlated? There's a cell-free and the cell-based, and how do we actually design this even experiments better? I think that is the key. That the phase 1. So when people say, well, there's no outcome or the results came out of that phase one. I don't look at it that way. I look at it as we learned how to actually interpret it or how to even design experiments on things like that, because we didn't know, right? So I think that opens up a lot of actually opportunity of experimental design, and it's a biological experimental design actually.
So we'll take 2 quick questions, please.
I don't know if any of these questions are entirely quick. John Balbus with NIEHS. And I have a similar question, but I'm going to take it down one level of organization. And I was very struck by your ontology. It was very interesting in that it goes all the way from the chemical structure and chemical space and then focused on apical endpoints. And it intersected at the level of the cell. And we just had a question about cellular ontology, but I'm going to go down one level and say that there's a little bit of a black box in there, which is where this committee has been focused a lot. We have a whole lot of incredibly massive sets of data related to protein expression, genetics, epigenetics, et cetera. I don't have to give the whole litany of all of the omics. And my question is, to what extent does your large-scale ontology get down and dig deep into that system and create an ontology to allow us to organize those data as well to do this kind of QSAR?
Long time ago, I've tried gene ontology against a chemical ontology, chemical structure ontology, and you could actually do that. You could actually interject the chemical classes into gene ontology and find those. But those are the times that our genomics experiment were not very reliable. But I think 15 years later, 10 years later now, I think we have much better chance of actually getting that worked out.
So before we take a question here, there's one online. How do you consider inorganics or organometallics in the 8200 compounds that are going to be screened in Tox21, and whether those will be amenable to QSAR approaches?
So that's a very, very good question and tough question, because QSAR historically, it has only been extended to or applied to organic chemistry. However, there are other methods that we could apply, not just QSAR. Okay, there are computational methods we could apply. And
thank you. And George, quickly, last
Sure.
question.
Thanks, by the way, Chihei. Really clever talk. It's about the cleft palate example that you put up, and I wasn't sure whether that was a QSAR or basically an expert rule-based system with multiple possible chemical fragments and rules that you did some sort of Bayesian analysis on. Which was it?
okay, so it actually all of the above. So the first thing I do is I need a data. I'm not a biologist, so I need a data. I could only learn from a data, right? So I learn from data, and then based on the data, I could actually build structural knowledge. Okay. And then I also learn biology, so I connect to the biologist. That's why it becomes a chemotype. It becomes a cleft palate chemotype when I actually assign this particular substructure with atomic properties like sigma charge positive or negative. That one could actually now be an inhibitor for this or agonist for that. Okay. So that is a separate learning that I'm doing. And based on that, once I have that, I could actually go into my larger database and find the chemicals that follow that. And I build training sets and then build QSAR.
Thanks.
Thank you.